Good evening. So today we'll begin with week nine's problem questions for NPTEL course Experimental Biotechnology. We'll start with the first question. The illumination of the specimen as well as the collection of the light is achieved by a single lens in dash. And four options are given here: bright field microscopy, phase contrast microscopy, epifluorescence microscopy, dark field microscopy. You can take a couple of minutes to think about this. And before we go on to the right answer, if you're aware of the different kinds of microscopy, the techniques, then we'll take it from there. uh, photo bleaching. So you have four options: anti-fading agent, buffer, antibody, and ethanol. You can take a couple of minutes to think about this and then we'll discuss. Okay, so before we go to this question's answer, does anyone here know what photo bleaching is? Okay, so let's say you have a fluorophore and you're exposing it to light. Now this will absorb photons. It will get excited, and when it is returning, it will emit fluorescence. But what happens if you use higher intensity that corresponds to higher intensity, not lower, not frequency or wavelength, higher intensity than what will minimally excite it. What this does is this it will maximally excited, that would lead to other reactions with free radicals and oxygen, and in such a way that the fluorophore will lose its ability to cause emission. Now, if this is the case, even if you try to excite it with a particular wavelength, it will not anymore fluoresce. So that is what you will be able to see in this figure. So if you see here in A, B, and C, you see decent fluorescence, but you will be able to see gradual decrease. Whereas in D, E, and F, you will be able to see clearly that the fluorescence from other regions, I mean, the green is also included. The GFP signal is also included. That's also constantly being decreased. So this is because again, the fluorophore is losing its ability to fluoresce. This is called photobleaching. Now, the question is which of these reagents will help protect the sample from high intensity laser beams to avoid photobleaching. So, uh, do you have any idea if the buffer will help or if there's any antibody? Okay. Are you aware of any anti-fading agents? Okay. No. What is the experiment that you did? Oh, you took silicon. Ah, okay. 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 I haven't physically done those experiments, but my knowledge here is based on what I've read. So we'll just go through the incorrect options. So the buffer is not going to help. The buffer will not be able to ensure that your fluorophore remains activated. And antibody here plays no role. Um, Usually, antibodies in different experiments are tagged with fluorescence producing molecule. Like, for example, in Western blots, you have your secondary antibody linked to an enzyme that would produce a product which is fluorescent. So that also has no role. Whereas your ethanol that used in fixation or dehydration. So that would not normally be helpful in improving preventing this bleaching. But we have these chemicals called as anti-fading agents, like this propyl gallate and DABCO, that people use in the mounting media to prevent the fluorescent dyes from photo bleaching. Right. So this is how photo bleaching looks. Okay. The next question is a microscope with a numerical aperture of zero point 0.3 and a light of wavelength 300 nanometer is used. The maximum resolution is dash. Yeah. So there's a formula to calculate this. Uh, Before you start with that, you can tell me what numerical aperture is and what wavelength is, and we'll talk about resolution and then be able to correlate these. You can take a couple of minutes to think about this. Okay, so numerical aperture, it tells you how much light can enter and how fine, at what resolution you'll be able to spot the images. Now, there's this one formula that relates your maximum resolution based on numerical aperture and the wavelength. This is called Abbe's resolution limit, and it just tells you that d, which is the resolution, is equal to lambda by 2 into Na. So when we have lambda is equal to 300 nanometers and numerical aperture is zero point 0.3, so 300 divided by zero point 0.3 into 2, so that will be 500. So the maximum resolution when we have a numerical aperture of zero point 0.3 and the light of wavelength 300 nanometer is 500 nanometers. Fine. Okay. The next question is, in scanning electron microscope, the dash are produced as the image. Option A, topological features. Option B, protein structure. Option C, bright field. Option D, dark field. Take a couple of minutes to think about this question and then we'll discuss. So first you can tell me what scanning electron microscope is and we'll take it from there. Okay. Do any of you know uh, how SEM works? Okay, you can tell me what purpose you are aware of. We'll take it from there. Morphology of. Okay, why not? To look at what is within, as in like inside. Okay, right, right. So yeah, so in scanning electron microscope, we, like you mentioned, we look at the morphology, and um, so we are talking about the surface of bacteria, very large surfaces. Will we be able to point out specific regions and tell this is where a protein is, this is how that protein looks like using scanning electron microscope? But is the resolution that good? So the proteins are on the order of few angstroms. Let's say 50 angstrom this side, 50 angstrom on all three axes. That is very small. That will be not even 1% of what you see on a microbe, on a bacteria, which is on the order of microns. So do you think with SEM we'll be able to get the protein structure? Okay. So this is where the concept of resolution comes in. So in SEM, like you mentioned, what happens is you have this electron beam that will touch the surface, and when it's hitting the surface, you have some secondary emitted electrons. Now, based on these emitted electrons, we get an idea about what are how is the surface present. As in, if you have any bulges on the surface, the secondary emitted electrons for that will be different from your flat surface. Based on this, you get an idea about the texture and the 3D features, which is what is called as the topology. But the problem here is the resolution at which we can do SEM is very, very uh, low, which means it's on the order of nanometers. So you'll not be able to typically talk about uh, protein structures, because for protein structures we have been routinely using methods like X-ray crystallography, which gives you at the angstrom level. So SEM will not work. And bright field and dark field, like we saw in the first question, these are light microscopy techniques. These have no relevance to scanning electron microscope. So the right answer for this question is in scanning electron microscope, the topological features are produced as the image. Right. The next question is dash is used as the electron gun in FE-SEM. And four options are given here: tungsten filament, LaB6 filament, crystal tungsten, crystal LaB6. Take a couple of minutes to think about this and then we'll discuss. Okay, so do you know what FESM is? Okay, no issues, no issues. So we already talked about scanning electron microscope. Now in field emission, in FESM, which is also expanded as field emission scanning electron microscope, the source for your electron is an electron gun that is a sharply pointed single crystal tungsten tip. Now, because this tip is sharply pointed, the strong electric field that is being applied there, it allows for field emission of electrons. This means the beam of electrons that we have will have very high brightness, which means, brightness, which also means that the spatial resolution that we get here is better compared to thermionic sources. Now, thermionic sources are what you saw here as the options, like for example, LaB6 filament and tungsten filament. In these filaments, like how normally you would have seen in other filaments, you just pass current through it. So the heat is what helps you generate the electrons. Yes, and that's what we have as the options here. The filaments here, like tungsten and LaB6, which is lanthanum hexaboride, these are thermionic emitters. Um, but this crystal tungsten tip is what we need for field emission. So this is basically a better method to be able to produce electrons with high brightness and small energy spread. So you get better resolution of your images. Right. The next question is dehydration of a sample is necessary before SEM imaging as water does dash. And four options are given here. It does not diffract electron. It diffracts electron, increases the background sample, produces only backscattered electrons, produces only Auger electron. Okay, you can think about this for a couple of minutes and then we'll discuss. Okay. Now, before we go on to dehydration, because a couple of answers here talk about diffraction. Do you know what diffraction is, or what is minimally needed for diffraction? Okay. Yes. Yes. Okay. And do you know where we apply it? I mean, that principle. Yeah, yeah, we apply it in many cases. Like we had discussed a few, I mean, at least we had come across a few terminologies in the previous slides, which make use of the diffraction property. There was this one thing called X-ray crystallography. So there, what we do is we try to grow crystals from proteins, and these protein crystals usually act as your diffraction grating. So there the concept of diffraction is important. When you put X-rays, it hits this protein crystal, and because of this diffraction and constructive interference and destructive interference, we get few spots with which we'll be able to trace back to the atoms and position of atoms. Right. So now with respect to SEM imaging, we do the dehydration and to remove that is to remove water. Now that said, uh, if we look at the four options here, does dehydration is necessary because water does not diffract electrons, or does it diffract electrons and increase background, or does it produce backscattered electron, or it produces only Auger electron? So if we look into this, um, in scanning electron microscope, first we have the specimen vacuum. So even if we have the sample with water, because in the low pressure, these the water on the sample, it will vaporize, and I mean it will vaporize non-uniformly in a way that it can damage the sample. And if there is this water, it will of course interfere with our sample by increasing the background noise. But what it does not do is it cannot diffract electrons. Um, and also it does not produce the backscattered electrons. So backscattered electrons are those which can differentially, what do I say, change the angle of your electrons. And for this, you need elements with higher atomic number, whereas with water, you cannot. And again, Auger electrons, these are generated by ionization of the inertial. But with the kind of wavelength that we use and the energy that we use with SEM, this is not possible. So the reason that we do this dehydration treatment uh, is because it can diffract the electrons and it can increase the sample. So earlier we had also mentioned about ethanol and fluorescence. Adding ethanol immediately removes the water, and this is routinely done in SEM. The next question is, to make the sample conductive, it is dash. Option A, coated with conductive material. Option B, coated with silica. Option C, coated with liquid nitrogen. Option D, carbon nanoparticles. You can take a couple of minutes to think about this and then we'll discuss. Okay, so in what conditions do we want to make a sample conductor? When would we want to make a sample conductor? Any, okay, okay. If we don't make it conductive, what will happen? Right, right. Good. So, yeah, like you mentioned, but a bit more specific here it's given. So, these non-conductive samples uh, will lead to image distortion. What you get as an image will not, you will get something, but it will be more of noise. So, usually they are coated with thin layer of metal or metal alloy like gold. People routinely do gold plating. You might have observed samples being plated with gold, or they also do with platinum and gold or palladium alloy. Uh, now, the thing is, when you do this, it makes it conductive. See, in SEM, what we're trying to do is we want to see if what we have, uh, uh, if the surface morphology could be captured. But if we So even if we coat it, as long as the thin coating that we have is evenly distributed, we'll be able to get the morphology. And it is conductive because we use these electrons. But if we coat it with silica, silica is nothing but your sand or glass. Silica is a component of both sand and glass. That would make it bad. In liquid nitrogen, we just use it for freezing. It has nothing to do with conductivity. By freezing, what I mean is, let's say you take a bacteria. You have worked with bacteria, right? Bacteria or some microorganism. That's what you did SEM with, right? Or you know that people do SEM with, right? Yeah. So let's say you give it different tests, like for example, hydrogen peroxide tests and so on. So in that cases, there will be changes in the bacterial morphology. So the thing is, when you add this, these changes happen in fractions of second. So to be able to capture what changes happen, you can freeze it. If you freeze it, now the morphology is frozen. Then you can use it. So with this coating with liquid nitrogen, it doesn't do anything with conductivity, but rather freezing the sample. Now carbon nanoparticles, like it is mentioned here, that carbon is used for making it conductive, but they don't mention about nanoparticles. And like we had mentioned earlier, you need, as long as you have a uniform coating, you'll be able to get the morphology back in SEM. Right. The next question is related. We discussed about what this coating is. Now, what is the name that people give for coating sample in SEM? Option A, sputtering. Option B, moisturizing. Option C, air drying. Option D, nanomaterialization. Sputtering. Okay, good. So, yeah, sputtering or sputter coating. This is again the method with which a layer of conductive material is coated onto your specimen. So this way, your once your electrons hit, your secondary electron emission is improved, and that way you will be able to get a good idea about the morphology. Now, moisturizing. In SEM, we want these dry samples, like we mentioned earlier. We do not want water, so moisturizing is a bad idea. Air drying. So this again involves removal of water molecules. This is a dehydration step, but has nothing to do with coating your conductive material. Now, nanomaterialization. Again, it's not relevant. Earlier, we had this discussion on whether carbon nanoparticles will be used, but it was not really the case. So, this process of coating your sample is called sputtering or sputter coating. Okay. The next question is dash is used as a staining reagent in transmission electron microscopy. This is 10. You can take a couple of minutes to think about this and then we'll discuss. Okay, so in TEM, we do staining of sample. Uh, so in TEM, the stains are with heavy metals. So they scatter the electrons and provide contrast. People use uranyl acetate, lead citrate. So one of these can be used as a staining reagent in TEM. Even for protein samples, we use TEM. And this is the method called cryo, cryo-EM, cryogenic electron microscopy, where TEM is the phenomenon that is being used. You take your protein samples, you put it on a plate called as grid, and you plunge it in liquid nitrogen. Then you use this stem. Here also, transmission electron microscopy can be used. And with this, you can obtain resolution of structures up to close to 2 angstrom. Fine. The next question is, the secondary antibody used for immunolabeling is coupled with dash, gold, silver, copper, and platinum. You can take a couple of minutes to think about this and then we'll discuss. So in immunolabeling, we use gold. So for TEM here, the secondary antibody is conjugated with colloidal gold particles. Now, why gold? So it is electron dense, which means the contrast will be higher, and people can synthesize this at different sizes. This is nothing but nanoparticle synthesis. And what about other metals? Silver, it can be used, but it is not directly conjugated to antibodies. Copper is used for TEM grids, what I had mentioned in the previous slide, not for antibody labeling. And platinum, it is used for coating enzyme, but not for antibody conjugation. Right. The next question is a biological question. Parasite culture is synchronized at ring stage by dash d sorbitol, DMSO, GMSA, organic solvent. You can take a couple of minutes to think about this and then we'll discuss. Okay, in parasite culture, let's take the case of Plasmodium falciparum. So you have different good stages, and synchronization at ring stage, it is done with sorbitol treatment. Sorbitol, do you know what sorbitol is? So let's say you take glucose, you reduce the aldehyde of the glucose into an alcohol, you'll get sorbitol. It's just a sugar with an alcohol. Now, when you do this, it acts as an osmolyte and it also lyses other stages like the trophozoite and schizoid infected red blood cells. But the ring stage parasites, these are intact. So this way we can ensure that all the ring stage parasites are only selected when you do a sorbitol treatment. So uh, this synchronization is done with sorbitol. And if you look at the other options, DMSO, which is dimethyl sulfoxide, we use this for dissolving compounds that are not compounds, or in this case, like it is shown as drugs that are not usually water soluble. DMSO tends to dissolve most of hydrophobic compounds. We use that routinely for crystallization. GMSA, that's a staining dye that is used to visualize these parasites. And organic solvent, it is DMSO is one of the organic solvents. The next question is to calculate the number of cells, dead and alive, DAS solution is used. Goldstein, trypan blue, bromophenol blue, xylene cyanon. You can take a couple of minutes to think about this. Okay, yes. So, what is trypan blue and how can it uh, differentiate dead and alive cells? Okay, right. So, similar to your propydium iodide, this is also dependent on the ability of the membranes to be intact or not. So, in live cells, the cell membrane is intact. So, trypan blue cannot enter. But in dead cells, these membranes are not intact, which is why it can take up the blue. Whereas Goldstein, we don't use it for cell viability. And bromophenol blue and xylene cyanon, these are tracking dye that people use for electrophoresis. Like, for example, what we learned earlier in SDS page and so on. Okay. This is a very, very basic immunology question. Kupffer cells are a part of DASH, lungs, liver, heart, and kidney. Yes, these are macrophages that are specifically in liver and they play a role in immune surveillance. In lungs, we have these alveolar macrophages, in heart, cardiac macrophages, and in kidney, we have mesangial cells. So, cells are specialized and present only in liver. Good. The next question is dash lasers are used for scanning in flow cytometry. Argon lasers, neon lasers, helium lasers, radon lasers. So, um, before we get into the kind of lasers, do you know what flow cytometry is? Okay, so flow cytometry is the method where we pass in these cells that contain a fluorescent marker, and uh, we will be able to segregate cells with gating. So here we have these argon ion lasers, and these are used for excitation of the fluorophore. Neon lasers are not used. Helium lasers can be used for certain fluorophores, but people generally use these argon lasers. And radon is radioactive. It is not inert. Can't be used for laser. The last question is gating is the dash selection of only a certain population of cells for analysis on plot, selection of one cell for analysis on plot, selection of preferred voltage for analysis on plot, selection of preferred wavelength for analysis on plot. This is again relevant to flow cytometry. Okay, so I told that in flow cytometry we can separate cells using fluorescence. So in this method, only certain cells with exhibit fluorescence have to be separated from cells that are not exhibiting fluorescence. So gating here means drawing a boundary, which tells us that this subpopulation of cells will be used. So the right answer here is gating is the selection of only a certain population of cells. Right. So with this, we come to an end for today's lecture. Uh, if you have any doubts, you can ask me. I'll stay on the call. So no, no, you just so this is just an introductory course. So if you know the differences between the basic microscope techniques, that should be more than enough. Like for example, if you had noticed here, we just looked at how light passes through, what is entering, what is exiting. Based on that, if you have a basic understanding, that should be more than enough for this course. And anyways, you will have components from other week's lectures as well. So I don't think you'll have to get in too deep into the concepts of this microscope. Having a basic understanding would be sufficient. So yeah, basic principle. Like for example, in scanning, electron, we measure the secondary electron, and based on which we reconstruct the surface. Whereas in transmission, we allow the electrons to enter the sample. So these, at least these differences, we should be aware of. If you're not aware of these differences, it will be difficult. So these you can focus on. Okay, thank you.